Thank you, Sandy, very much, and greetings from across the bay in Berkeley. It's a pleasure to help celebrate the Biohub's 5th anniversary by sharing some of the work in my lab that the Biohub has enabled. Unlike most research funding, the Investigator program encourages and supports risk-taking. For me, that meant using our biophysical perspective and experimental methods to explore new directions, including how viruses physically interact with cells. While I've been an unwitting host for many viruses over the years, I had not had the chance to explore them scientifically until the Biohub made that possible. Today, I'll tell you about 2 new ways that viruses have evolved to overcome host barriers to infection. One virus I'll discuss is influenza A on the left, which we discovered can move in a directed and persistent manner thanks to the organization of its envelope proteins. The second is a reovirus on the right, which we found hijacks the host cytoskeleton to drive cell-cell fusion by locally nucleating actin assembly. Next slide. Let's start by taking a step back. Viruses are physical objects that must overcome physical barriers as they bind and enter cells, replicate and release their progeny, and start the cycle of infection all over again. These physical barriers can include cell membranes, mucus layers that limit the entry of material into cells, as well as the host immune system that attempts to kill infected cells and the viruses themselves. Hence, viruses must outsmart host defenses with small genomes and a limited repertoire of proteins, a real David versus Goliath battle. Now, while viruses can and do play the numbers game, that is, make lots of progeny and hope some succeed, we are finding that some viruses are able to cleverly organize themselves and their surroundings to increase the chances of success. Next slide. In the case of influenza A, like other respiratory viruses, a major barrier is penetrating the mucus layer atop airway epithelial cells before mucociliary transport clears the virus from the airway, thereby protecting the underlying epithelial cells from infection. Influenza A must overcome this barrier and make its way to a host cell to infect by binding to sialic acid, its receptor, a sugar found in mucus and on many cell surface proteins. Next slide. To make its way through the mucus and into cells, influenza forms virus particles that encapsulate its RNA genome and are surrounded by a membrane envelope. The envelope contains transmembrane proteins that are essential for viral entry into cells, the 2 most abundant of which are hemagglutinin, or HA, which binds to sialic acid, and neuraminidase, or Na, which cleaves sialic acid. Having both is important. If the virus could only bind to sialic acid, it would get stuck in the mucus and be swept away. If the virus could only cut sialic acid, it would never be able to hold on to and enter cells. So having both on the envelope is important. Next slide. So what is the optimal amount of hemagglutinin and neuraminidase on a viral envelope? Well, Mike Vahey, the postdoctoral fellow who led this project and developed strains of influenza that could be fluorescently labeled to quantify proteins at the single virion level, found that there really isn't a single optimal ratio. Rather, influenza A encodes phenotypic diversity in the progeny from each infected cells, both in terms of the ratio of envelope proteins and in terms of virion size. In these fluorescence images shown in this slide, where hemagglutinin, HA, neuraminidase, Na, and the nucleoprotein are all labeled, you can see the variation in protein amounts and in virus shape, with some virions appearing to be spherical and others filamentous with lengths up to 10 microns or longer. This biochemical and geometrical heterogeneity allows the virus to escape dramatic population bottlenecks during infection. Next slide. What about the organization of these 2 envelope proteins, HA and Na? Is it important that they be organized in a certain way for entry? Interestingly, the HA and Na on the surface of a virion are anchored in place and do not diffuse around like many cell surface proteins do. In these photo-bleaching experiments shown on the left, fluorescently labeled HA and Na on filamentous virions do not reorganize on timescales of tens of minutes. When Mike used super resolution microscopy to quantify the organization of HA and Na, he found that they were asymmetrically distributed, with Na on one end and HA on the other end of the virus. Because there is no diffusion, the spatially separated regions of HA and Na remain that way as the virus encounters mucus and cell surfaces. Next slide. So could this asymmetry in binding and cutting activities be useful to the virus? To answer that question, Mike devised a clever in vitro experiment where influenza A was dropped onto a surface and imaged over time. Remarkably, the virions did not diffuse randomly. Instead, they showed directed and persistent movement, a surprising observation given that viruses are not normally thought of as being motile. You can see in this video on the left that the virions move away from the end with Na clusters and towards the end enriched in hemagglutinin. They're also more persistently moving along the long axis of the virion rather than in the direction of the short axis. Next slide. To test whether asymmetric binding and cutting was all that was needed to create and directed and persistent motion, rather than more common forms of nanoscale motion like molecular motors, Mike set up a Monte Carlo simulation that included only those features, binding and cutting, together with thermal motion. Sure enough, he found that asymmetry in HA and Na led to motion of the virus. Essentially, the neuraminidase cut sialic acid at one end of the virus so that when the virus randomly unbinds and binds again due to thermal forces, it has no choice but to step in the direction of available sialic acid and away from the area that has been cleared of sialic acid. The elongated shape of filamentous virions, typically found in clinical isolates, limits rotational diffusion, causing preferential motion along the axis of the virus. While these are only 2D simulations, we also looked experimentally at the path of influenza A in 3D mucus formed atop epithelial cells and found that the directional tracks were visible there as well. This suggests that by spatially organizing its envelope proteins, influenza A may be able to more efficiently penetrate airway mucus layers using directed and persistent motion rather than just random diffusion, enabling it to reach the underlying airway epithelial cells before the mucociliary transport carries it away. Next slide. Thanks to the Biohub's joint postdoctoral fellow program, I'm pleased to welcome Dr. Sandansh Agarwal, who is now anchoring a collaboration with the groups of Greg Huber and Shailin Mehta at the Biohub to further explore how diverse viruses navigate the physical complexities of the airway. Next slide. Now I'd like to switch to the second virus, a reovirus, which has evolved a different but equally clever way to successfully avoid host defenses. The reovirus strategy is to drive infected cells to fuse with their neighboring cells. This allows the virus to remain inside and hidden from host immune cells while more virus particles are produced. Next slide. Reoviruses are taken up by cells through endocytosis, and once inside, express a protein known as a fusion-associated small transmembrane, or FAST protein, that causes the neighboring uninfected cells to fuse and form multinucleated syncytium. The fluorescent images on the left show two cells expressing a FAST protein in purple that merge with surrounding cells over time to form much larger and multinucleated cells that continue to express the fusogen. Over even longer periods of time, shown in this video, the process of cell-cell fusion can merge many cells into a rather menacing syncytium that moves as it absorbs additional cells. And this will continue as the cells fuse and eventually rupture, releasing the virus. But the larger cytoplasm allows the virus to continue replicating before it does this release. Next slide. The specific FAST protein responsible for the cell-cell fusion shown in the previous slide is called P14. And it looks nothing like other known fusogens, which are tall and undergo multi-step conformational changes to force two membranes into close contact. Now, Carmen Chan, a grad student in my lab who led this work and became a FAST protein expert, was interested in how this 125 amino acid long protein with no obvious structure could accomplish the same thing as these much more complex viral and cell-cell fusogens? To answer that question, she focused specifically on the cytoplasmic tail. Now, we know that cutting off the cytoplasmic tail of P14 prevents cell-cell fusion. So what is this short peptide on the intracellular side of the cell doing to help with cell-cell fusion? Next slide. Through a combination of computational analysis and experimental work, Carmen first showed that drugs that disrupt the actin cytoskeleton, and specifically the formation of branched actin networks, also attenuated cell-cell fusion. She then identified a binding site on the cytoplasmic tail of P14 that bound the adapter protein called GRAB2, which Carmen then showed in live cells and in biochemical reconstitution specifically triggers the nucleation of branched actin networks. As shown in the plots on the right, mutations to the GRAB2 binding site disrupt actin assembly and abrogate cell-cell fusion. Next slide. Much like how nucleation of branched actin networks are used by motile cells to extend their leading edge, or by macrophages to phagocytose target particles, the P14 fusogen appears to hijack the actin cytoskeleton of host cells in order to locally push two membranes into close contact, where the short ectodomain of the fusogen can then promote membrane mixin and full fusion. We hypothesized that the force generated by actin is required because of the plasma membrane is crowded with proteins that keeps the lipid bilayers physically apart and separated from each other. So driving fusion thus requires overcoming that plasma membrane protein barrier in order for the membranes themselves to merge. Next slide. Now, P14 is not the only small fusogen in the reovirus family. Indeed, there are at least 4 more that appear to be very similar, short, no structure. So the obvious question is, do these other fast proteins contain the same adapter protein binding site that lets them hijack the actin cytoskeleton to drive cell-cell fusion? And the answer is no. So Carmen and collaborators from the physiology course at the Marine Biological Laboratory set out to examine whether a divergent fusogen, P22, which is separated from P14 by about 500 million years of evolution, whether P22 also requires actin to drive cell-cell fusion. And if so, how? Next slide. Through a similar combination of computational and experimental work, the team found that P22 does indeed use branched actin network assembly to drive cell-cell fusion, but it does so through a distinct pathway that depends on intersectin rather than this other adapter protein GRAB2. Moreover, we found that these fast proteins are highly modular with ectodomains and cytoplasmic tails that can be swapped. Indeed, we could create a superfusogen by combining the P14 ectodomain and transmembrane domain with the P22 cytoplasmic domain, which was more fusogenic than either P14 or P22 alone. So we conclude that these reovirus proteins and possibly other poorly understood cell-cell fusogens that are currently under study require actin-generated forces to bring two membranes close together for cell-cell fusion, but that diverse actin nucleation pathways can be used to generate those forces. Next slide. So what I've shared with you today are 2 new ways that viruses can overcome host defenses. One, influenza A, by spatially organizing its envelope proteins to generate directed and persistent movement. And another, a reovirus, that works by spatially organizing the host cell actin cytoskeleton to drive cell-cell fusion and give itself more space to replicate. In both cases, the viruses have figured out ways to overcome physical barriers imposed by the host, specifically those physical barriers created by crowded cell surfaces and secreted protein networks, materials whose physical and biochemical properties are still not well understood, but affect not only pathogen entry, but things like tissue formation and communication between cells. While the examples I described here are only 2 of many, many strategies that viruses have evolved in the age-old battle between host and pathogen, understanding the mechanisms needed for successful infection will hopefully bring us closer to finding new ways to defend against them. Next slide. I want to thank my wonderful lab for being collaborative and fearless scientists and engineers, particularly the two whose work I described in this talk. Carmen Chan, who is now a postdoc at Harvard, and Mike Vahey, who now has his own lab at WashU St. Louis. I'm also grateful to the Chan Zuckerberg Biohub for their generous support, and to Joe, Steve, Bill, Sandy, Gauss, and so many others for creating a wonderful Biohub community. Not to mention providing excellent lunches, snacks, and copious coffee at the Biohub, which no doubt accelerates research. Next slide. Thank you, and I'd be happy to take any questions.
Dan, so Matthew Akamatsu asked, do either of the fusogen proteins also trigger additional membrane remodeling, exocytosis or endocytosis?
That's a good question. We haven't looked expressly at that. The cells that are expressing these fusogenic proteins do have disrupted behavior. So it would make sense that wherever this cytoplasmic tail is present and the protein is expressed and decorated all around the plasma membrane, that it would serve as small nucleation sites for other branched actin networks. And whether those then interfere with endocytosis, exocytosis, other processes that rely on branched networks, that's a really interesting question and something that we should look into further.
That's great. And sort of in the spirit of your really thinking about how physical forces work on the very small local level, there's a question from Rebecca Heiss, which is, what is the influence of host cell stretch on the fusion events? For lung infection, these cells are undergoing cyclic deformation.
Yeah, really interesting question. So can changing the stress or the tension in membranes influence the fusogenic process? I think it will have an effect in two ways. One, the higher the tension in the membrane, the more challenging it is to deform that membrane to bring it into close contact with an opposing membrane. However, an increase in tension helps to open fusion pores once they form. And so it can be beneficial and hurtful at the same time. And so it's interesting to think about, are there particular states of the cell, physical states of the cell, that are more susceptible to this fusogenic process than others? And that's another great question to pursue.
So, and then stepping back, a general question. So, before this, we may not really have thought, well, maybe people have, but thought much about how viruses, airway viruses cross the mucosal barrier. So for viruses like coronavirus or SARS-CoV-2 that may not have clear enzymatic activity in cell surface proteins, have you or others begun to look at whether there is also directed movement across a mucosal barrier?
So we haven't looked at that explicitly, but I think that is going to be one of the considerations for the success of the virus. One of the challenges of influenza, if it is a larger virus, is that its diffusive rate is going to be slower. So for smaller viruses, being able to diffuse is more of a reasonable solution to getting through the mucosal layer. But I don't know of any evidence yet of motile active motility in SARS-CoV-2, but understanding, do they simply take advantage of a breakdown in the mucus or a thinner layer of mucus? Do they simply play the numbers game better of having many particles and hoping some succeed? That's a great question for the coming years of the Biohub.
Another question from Adrian Jacobo. What is the advantage for influenza viruses to be able to move in a straight line? Are there gradients that might guide them towards cells?
Very good question. There doesn't appear to be a specific kind of gradient that the virus would be sensitive to in the mucosal layers on airway epithelial cells. But the advantage of moving in a straight line is that even if you're not moving towards a particular source or signal, it allows you over a fixed period of time to get further away from your starting point. So if you imagine a virus on the surface of a mucosal layer, if it set out directed movement in all different directions, at least one of those virions is going to be successfully able to cross through and find an airway epithelial cell, even if it's not guided by a gradient. So it still is advantageous, although it would be even more efficient if a gradient were influencing it. And possibly a gradient in sialic acid or in density of mucins could influence that process. And that's a good direction to explore.
That's great. Now, one thing that's really striking about the reovirus case is, I mean, as you know, there are other pathogens like bacterial pathogens such as Listeria that also take advantage of being able to mobilize the actin cytoskeleton for, so they are intracellular pathogens and mobilize the actin cytoskeleton for cell-to-cell spread. Is there any clear relationship between how Listeria is propelling itself through the cytoplasm and into an adjacent cell, and the mechanism involving the reoviruses and FAST?
Yes, there's a connection there, and that is in providing a way to trigger branched actin network assembly in the case of the bacterium on the bacterial cell surface, or in the case of the reovirus on the plasma membrane. And so the connection there with Listeria is the production of ACT-A, which is able to drive nucleation of branch networks. In the reovirus case, it has to work a little bit further downstream. It needs to be able to bind an adapter protein that then recruits the host cell machinery that is normally responsible at the plasma membrane for generating branch networks. And I had the pleasure to work on aspects of Listeria in my postdoc with Julie Theriault at Stanford. And so the analogy there between a bacterial pathogen getting pushed from cell to cell, and now evidence that a virus can take the same strategy of not having to package all the material to generate force, but really just intercept a pathway that already is very robust and active in generating force, and use that for its own purpose to bring two membranes together.